Hi everyone, this is Reema, your biology educator from V-Learning. So as you know that V-Learning is an online educational platform where we provide quality education for all. So to get all the benefits from our channel, do subscribe to it, and also don't forget to visit our website, that is www.v-learning.in, where you will be getting all the pre-recorded videos, the live classes, animated videos, and also the PDF notes of all these topics. So let's begin today's class. So you remember in our last class we have started with your chapter number 14, right? So where you have learned about the respiration in plants, and you have seen that there are two types of respiration, aerobic and anaerobic respiration. So in the last class you have seen that the pathways, that is which is required for the breakdown of the food in case of the anaerobic respiration, that is glycolysis. You have studied the various pathways of the glycolysis, right? So in today's class you will be learning about the various pathways in case of the aerobic respiration. Okay, that is, you have seen that the end product of glycolysis is the pyruvate, right? So the pyruvate is being further oxidized in the mitochondria, okay, and which is used for the synthesis of the ATP. So today you will be learning about the Krebs cycle and oxidative phosphorylation, okay, and this is your lecture number 2. So let's begin today's session. So see, so what is this Krebs cycle and electron transport chain? Okay, electron transport chain is also known as oxidative phosphorylation. Okay, so where does this take place? It takes place in the mitochondria of the cell. Okay, you know that mitochondria is the powerhouse of the cell, right? So the Krebs cycle and the electron transport chain, everything takes place in the mitochondria of the cell, and this electron transport chain is related with the Krebs cycle, because whatever the electrons that will be released, that is NADH2 and FADH2, will be used for the electron transport, will be used for the synthesis of the ATP in the electron transport chain, that is oxidative phosphorylation. Okay, that is oxidation of electron will take place basically. Okay, electrons will be transferred from various protein complexes. So inside the mitochondria there are various protein complexes where the transfer of electrons, that is oxidation of electron, will be taking place. Okay, so we will get the end product, that is how the ATP is being synthesized by the oxidation of the electron. So first we will learn the Krebs cycle, then we will look into the oxidative phosphorylation or the electron transport chain. So as you know that the Krebs cycle, where it takes place, it takes place in the mitochondria. Okay, in the mitochondria of the cell. Now, this Krebs cycle was discovered by Hans Krebs. Okay, so how the name Krebs cycle came is it was discovered by Krebs, that is Hans Krebs. Now, what is this Krebs cycle? It involves the oxidative metabolization of acetyl units and serves as the main source of energy. So, what it involves? It involves, that is, it is a process basically where the oxidation of the electron is taking place. Okay, so oxidative metabolism of the acetyl units and serves as the main source of energy. That is, oxidation will take place, and from that we will get, that is ATP will be synthesized. Okay, now it is also known as Tca cycle or citric acid cycle. So what is the other name of Krebs cycle? The other name of Krebs cycle is Tca cycle or citric acid cycle. Clear? Now, why it is known as Krebs cycle? Because it is named after the scientist who has discovered this cycle. So, he is the scientist, okay, he is the scientist who has discovered this cycle. So, after his name only, that is, it is called as Krebs cycle. Okay, his name was Hans Krebs, right? Because he has identified the various steps involved in the citric acid cycle. So in the citric acid cycle there are total 9 steps involved. Okay, there are total how many steps? There are total 9 steps involved. In the glycolysis pathway you have seen that there are total 10 steps involved, right? How the glucose is being broken down to pyruvate, right? But in case of this, 9 steps are involved, and it is a cyclic process. It will continue. Okay, now see, next, Next, it occurs in the matrix of the mitochondria. So where does it occurs? It occurs in the matrix of the mitochondria, that is in the inner membrane of the mitochondria. So which one is the matrix of the mitochondria? So see, mitochondria has got two membrane, right? One is outer membrane and the inner membrane. So the inner membrane, that is, these are the cristae, right? These are the cristae. So the Krebs cycle, it takes place here in the matrix, okay, in the matrix. It takes place in the matrix. All right. So it takes place in the cristae. Okay, these finger-like projections which you see, what is this called as cristae, right? Cristae. Yes, so here various F0 and F1 particles are there. Okay, here various, what happens? F0 and F1 particles are there, which helps in the synthesis of the ATP from the ADP. All right. Now see, it occurs, yes, in the matrix of the mitochondria. Next, as you have seen that there are how many steps? There are 9 steps. There are total 9 steps involved in this process. And what is being formed? NADH and FADH. Okay, what are the products? NADH and the FADH. These molecules are being formed during the Krebs cycle, okay, when oxidation takes place. Now see what happens in the Krebs cycle. So pyruvate, as you have seen that in the glycolysis, what happens? In case of glycolysis, the end product is what? The end product is pyruvate, right? Pyruvate. In case of the anaerobic respiration, this pyruvate also forms what? This pyruvate also forms lactic acid, right? Lactic acid, that is when it is formed in the muscle, isn't it? And also this pyruvate is also responsible for the formation of what? For the formation of carbon dioxide and ethanol, which is used for the production of what? Production of the there is fermentation of yeast. Okay, so in case of the anaerobic respiration, what you have seen in case of anaerobic respiration, the pyruvate which is being formed here, the end product of the glycolysis, the pyruvate, it got converted further into lactic acid, right? It got further converted into lactic acid. Whereas in case of aerobic respiration, the pyruvate gets converted into acetyl coenzyme, okay? So in case of aerobic respiration, which is taking place in the Krebs cycle, which is taking place in the mitochondria of the cell, okay? It is called as what? It's called as Krebs cycle. So in case of aerobic respiration, what happens? The pyruvate which is being formed here, okay, it will get converted into what? It will get converted into acetyl coenzyme A, okay, which is also a 3-carbon molecule. All right. So which is also a 3-carbon molecule. So this pyruvate is a 3-carbon molecule. So during this process, what happens? During this process, one of the carbon dioxide will be lost. Okay, one of the carbon dioxide will be lost, and it will form what? It will form 2 carbon molecules. So let us see what happens during this process. So the pyruvate which is being formed, okay, pyruvate is being formed from what? From the glycolysis, okay, from the glycolysis the pyruvate is being formed, right? So pyruvate is oxidized into what? Is oxidized into acetyl coenzyme. So during the process of oxidation, what happens? One of the carbon dioxide, that is decarboxylation, takes place. Okay, one of the carbon dioxide gets released out, and it leads to the formation of 2 carbon molecule. Okay, now what happens is this acetyl coenzyme A, it will combine with the oxaloacetic acid. OAA. Okay, oxaloacetic acid is a 4-carbon molecule. So acetyl coenzyme A combining with the oxaloacetic acid, it leads to the formation of what? It leads to the formation of citrate. It leads to the formation of what? It leads to the formation of citrate. Now here the enzyme involved is citrate synthesis. Citrate synthesis. Okay, the enzyme involved is citrate synthesis. Now this citrate will be further oxidized. Citrate will be further oxidized into isocitrate. Okay, isocitrate. So the enzyme involved in the oxidation of the citrate to isocitrate is aldolase. Okay, it is aldolase. Now, here what happens, further isomerization reaction takes place. So, the conversion of isocitrate to citrate and citrate is a irreversible reaction. Okay, it is a irreversible reaction. That is, isocitrate convert back to the citrate and citrate can convert back to isocitrate. So, it is a irreversible reaction. So, here what happens, hydration and dehydration together it takes place. Okay, two reaction takes place here, both hydration and dehydration takes place. Now isocitrate which is being formed, right, it gets further converted into alpha ketoglutarate. Okay, so during this process one of the carbon dioxide is being decarboxylation is being taking place. One of the carbon dioxide is being removed. Okay, that is reduction of carbon dioxide is taking place. So it is a 6-carbon molecule, right? It is a 6-carbon molecule. Now one of the carbon dioxide is being reduced. So what happens here, 5-carbon molecule is being formed. So alpha ketoglutarate is a 5-carbon molecule. So it is here the enzyme involved is isomerase. Okay, now alpha ketoglutarate is further converted into succinyl coenzyme A. Okay, further converted into succinyl coenzyme A. So here the enzyme involved is alpha keto dehydrogenase. So here the enzyme involved is alpha keto dehydrogenase. Now further the succinyl coenzyme A gets converted into succinate. Okay, succinate. Now the succinate gets oxidized into fumarate. Now from fumarate it forms malate, and again oxaloacetic acid is regenerated. So here total 9 steps are involved in the process of this Krebs cycle. In your book it is not given properly. Okay, in your NCERT it is not given, all the 9 cycles are not shown properly. Okay, so here only succinic acid is being formed. So here before that there is another step, that is succinyl coenzyme A. From the succinyl coenzyme A only it is being formed. So during this process when alpha ketoglutarate got converted, right, so here what happens, here one molecule of NADPH2 is being evolved. Okay, NADPH2 is evolved. Okay, and during the process of conversion of this succinyl coenzyme A to succinate, here also GTP is being formed. All right, so yes, look into this diagram. Okay, the citric acid, after that here is isocitrate, okay, then alpha ketoglutarate. So during the conversion of isocitrate to alpha ketoglutarate, NADPH plus H2 is being formed. Okay, now next the alpha ketoglutarate got converted. So during this process one more NADH plus H plus is being formed. Okay, now when alpha ketoglutarate that is succinyl coenzyme A got converted into succinate, during this process GTP is being evolved, right? GTP is being evolved during this step. Okay, next what happens, from the succinate then fumarate is being formed. Okay, here one more will come, that is fumarate. Then from fumarate acid or malic acid is being formed, that is malate is being formed. Okay, next again it is regenerated into oxaloacetic acid. So here one more NADPH molecule is being evolved. Okay, so this is how the cycle continues. This cycle it continues. Okay, again here this malic acid is leading to the regeneration of oxaloacetic acids. So this cycle it continues. Okay, it continues. So it is a cyclic process. Clear? Yes. So next, see the steps. Okay, step 1, the formation of citrate. So you have seen that the formation of citrate is taking place by the condensation, isn't it? So here look into it. The acetyl coenzyme A, it is what? It is combining with oxaloacetic acid. OAA is oxaloacetic acid, which is a 4-carbon molecule. So it combines, the acetyl coenzyme it combines with oxaloacetic acid and leads to the formation of what? Leads to the formation of citrate. Is that clear? Now next step is what? Next step is formation of isocitrate, right? So from the citrate, isocitrate is being formed. So here isomerization reaction is taking place. Okay, then formation of alpha ketoglutarate. So here oxidation is taking place, that is, so see here oxidation took place, so alpha ketoglutarate is being formed, right? So which is a 5-carbon molecule, one of the carbon dioxide is being lost, right? So next from the alpha ketoglutarate what is being formed? Succinyl coenzyme A, right? Then from the succinyl coenzyme A, succinate is being formed, then from succinate fumarate, then from fumarate malate, and again malate is regenerated to oxaloacetic acid, right? So this is very, very simple. So first what happens? First the pyruvate it forms acetyl coenzyme A, right? One of the carbon dioxide is released in the air, so it is a 2-carbon molecule, then it will combine with oxaloacetic acid, right? So combination of acetyl coenzyme A with the oxaloacetic acid, which is a 6-carbon molecule, it leads to the formation of what? It leads to the formation of citrate, right? So from citrate what is being formed? From citrate isocitrate is being formed, right? So from isocitrate what happens here? Isomerization reaction took place, right? Here isomerization reaction took place, isn't it? Now from isocitrate here oxidation is taking place, right? Here oxidation takes place, so it leads to the formation of a 5-carbon molecule, that is alpha ketoglutarate, isn't it? Which is a 5-carbon molecule. So alpha ketoglutarate. So further what happens? Further it is oxidized into succinyl coenzyme A, right? Then further succinyl coenzyme A gets converted into succinate. Okay, succinate. Then from succinate what is being formed? From succinate fumarate is being formed, right? Fumarate, right? So fumarate gets further oxidized into malate or malic acid, and malate is regenerated into oxaloacetic acid. So here regeneration is taking place, right? So that is together how many steps are involved? 9 steps are involved in the Krebs cycle. So this is all about it, and here various enzymes are also helping in the conversion process. Clear? Yes. So next, see, the Tca cycle starts with the condensation of the acetyl group with oxaloacetic acid. So what happens? The acetyl group, it will condense with the oxaloacetic acid, and it will lead to the formation of what? It will lead to the formation of citrate or citric acid, right? Then the reaction is catalyzed by the enzyme citrate synthesis. So conversion of the oxaloacetate and the to citrate, it is done by the enzyme citrate synthesis. Okay, and the molecule of coenzyme A is being released out. So the citrate is then isomerized to isocitrate, right? So the citrate is being isomerized, right, to isocitrate, right? Next, it is followed by 2 successive steps of Decarboxylation. Decarboxylation means removal of the carbon dioxide. Okay. Next, leading to the formation of alpha ketogluteric acid and then succinyl coenzyme A. So in both the process, what happens in both the process, one of the carbon dioxide is being lost. So here also carbon dioxide is being lost. That is decarboxylation taking place. Here also decarboxylation is being taken, right? Next, what happens? Next, during the conversion, yes, of acetyl coenzyme A to succinic acid, a molecule of GTP is synthesized. So when succinyl coenzyme A is converted into succinic acid, what happens in this step? In this step, a GTP molecule is being released. So this is succinyl coenzyme A, right? So it is getting converted into succinate. So during this process, what is being formed? GTP is being formed from GDP. Okay, GTP is being formed from GDP. So next, this is a substrate level phosphorylation. So this is a substrate level phosphorylation. In a coupled reaction, GTP is converted into GDP with a simultaneous synthesis of ATP from ADP. So GDP gets converted into what? GDP gets converted into GTP, and from ADP, ATP is being synthesized. Okay. Next, also there are 3 points in the cycle where NAD plus is reduced to NADH plus and H plus, and 1 point where FAD plus is reduced to FADH2. So see here in this cycle what happens. So see here 3 steps are there, right? So in 3 cases you will see that 3 times. So here what happens, from NAD plus it is being reduced to NADH plus and H plus. So here also when alpha ketoglutarate got converted, NAD plus got converted into NADH plus and H plus. So during the conversion, during the regeneration of the malic acid also, NAD plus got converted into NADH plus and H plus. So there are 3 places, where NAD plus got converted into what? NADH plus and H plus, and at one place FAD plus got converted into FADH2. So when fumarate got converted into malic acid, at that time when fumarate got converted into malic acid, at that time FAD plus got converted into FADH2. Okay, so yes. Next, this continued oxidation of acetyl coenzyme via the Tca cycle requires the continued replacement of oxaloacetic acid, the first number of the cycle. So it requires continuous replacement of the oxaloacetic cycle because it will be continued, right? In addition to it, it also requires regeneration of the NAD plus and FAD plus from NADH and FADH2 respectively. So for continuation of this Krebs cycle, it requires this molecule, that is NADH2 and FADH2. It is very much necessary for the continuation of this Krebs cycle. Now this NADH2 and FADH2 which is formed in the Krebs cycle, which is formed in the matrix of the Krebs cycle, is used in the synthesis of ATP. Okay, is used in the synthesis of ATP, which you will see in the electron transport chain, that is oxidative phosphorylation. Okay, this NADH2 and FADH2, that is the electrons, will be what happens, will be transferred here. Okay, will be transferred between the various complexes. Okay, so here it will lead to the formation of what? Ultimately we need the ATP, ultimately we need the synthesis of ATP. So it will take place in the electron transport chain. Okay, we will study in the next topic. So yes, now let us learn about the electron transport chain. So far you have discussed about the Krebs cycle. I hope you have understood what is being discussed, that Krebs cycle, here 9 steps are being involved, and the energy, that is the energy molecule or the electrons which are being evolved, that is NADH2 and FADH2. Okay, you have seen, right? Further, it will be used in the synthesis of ATP in the electron transport chain, which you will study now. Okay, clear? Okay. The metabolic pathway through which the electron passes from one carrier to another is called the electron transport chain. Okay, so it is basically a pathway, okay, where the electrons are being transported from one channel to another channel. Okay, so inside the matrix of the mitochondria there are various protein complexes. Okay, what are there? There are various protein complexes like complex 1, complex 2, complex 3, 4 and 5. So these electrons are being transferred through these various protein complexes. Okay, so it takes place in the inner mitochondrial membrane. So where does it takes place? It takes place in the inner mitochondrial membrane. So Krebs cycle, it is also taking place in the mitochondria, in the inner membrane of the mitochondria. Even the electron transport chain also, it takes place in the inner mitochondrial membrane. So see, yeah, now the mitochondria, it is a how many membrane are there? It is a double layered membrane, right? Double layered cell organelle. So it has got outer membrane and inner membrane. So this is the outer membrane of mitochondria and this is the inner membrane, right? Now inside the inner membrane, you know that there are various finger-like projections or folding, you will see, okay, various foldings are there, right? These foldings are what they are known as? They are known as cristae, isn't it? Cristae. Now this area is known as the matrix of the mitochondria. So in the inner membrane of the mitochondria, various that is ribosomes, okay, then DNA, all these things are present in the inner membrane of the mitochondria. Now the Krebs cycle and the electron transport chain, it takes place in the matrix of the mitochondria, in the inner membrane, that is in the matrix of the mitochondria. Now see here in the Krebs cycle, what is being evolved? We got NADH2 and FADH2, isn't it? This NADH2 and FADH2 is further used for the synthesis of what I said, that is ATP, right? Finally, we need what? We need only ATP, right? ATP should be synthesized, that is the energy molecule. So for that, inside the inner membrane, there are various electron transport chain. There are various proteins, okay? There are various protein complexes, okay? There are various protein complexes, okay? These protein complexes, what they do, they transport the electrons from one complex to another. So inside the inner membrane, there are various protein complexes. So they transport the electrons from one complex to another, okay? Now see, let us understand what are the various complex. So if this is the outer membrane of the mitochondria. Now, here what happens? This is the inside, okay? Here various protein complexes like complex 1, then complex 3, okay? Then complex 2, complex 4 and complex 5. All right. Now see, this is the matrix of the mitochondria. So here what is present, here we have learned that Krebs cycle, right? So during the process of Krebs cycle, what we got, what energy molecule we got? We got NADH2, right? And also we got FADH2, isn't it? These are the molecule which we got from the Krebs cycle. So further, these molecules will be that is will be getting excited, okay, and the electrons will be transported through these various complexes. So what is this complex? It is complex number 1, then this is complex number 3, this is complex number 2, this is complex number 4, and this is complex number 5. Clear? Now next, so NADPH which is being formed here, it leads to the production of FMN, okay, FMN. Now from this FMN, further electrons will be getting excited and it will form FES, okay. Now during this process, hydrogen which is being evolved here because of this energy molecule, this hydrogen will be released outside, okay, in the outer membrane. So this hydrogen will be released here. Now from this FES, okay, here there is another molecule which is known as ubiquinone, okay, I'm writing Q. What is that? Ubiquinone. Ubiquinone, right? Next, further ubiquinone gets converted into ubiquinone H2, right? Next, ubiquinone H2 leads to the formation of the electrons are further getting excited and converted into cytochrome B complex. Now from cytochrome B, it forms FES. Further, the electrons are transferred into FES, then cytochrome C1. Okay. Now here in the inner membrane, cytochrome complex is there. Okay. Now from the cytochrome complex, the electrons will be further transferred into, yes, further transferred into cytochrome A and then finally to cytochrome A3. Okay, this is how the electron transportation chain is taking place. Okay, electron transportation is taking place through these various complexes in the mitochondria. Okay, now NADPH, okay, is being excited and it transfers the various electrons, okay, to the complex 3. Now here in the complex 2, the FADH, it will form FES, okay. Now from FES it will form ubiquinone, then ubiquinone H2, and it will be sending back to the complex 3 here again, okay. Now during this process, the hydrogen which is being released here, all the hydrogen, it will be sent back to the complex 5, okay. All the hydrogen which is being thrown into the outer membrane, it will be collected and it will occupy in the complex 5, okay. Here in the complex 5, there is oxygen, okay. What is present in the complex 5? There is oxygen. So there are two particles, one is F0 and another one is F1 particle. So these two, F0 and F1 particle, collects all the hydrogen and throws it outside the cell. Okay. Now in this process, F0 and F1 particle, they used what? They used the ATP, that is they use ADP and convert into ATP with the help of the enzyme ATP synthesis. So ADP is finally converted into ATP here in the complex 5, okay. In the complex 5, finally the ADP is converted into ATP with the help of what? ADP is converted into ATP with the help of ATP synthesis enzyme, okay, synthesis enzyme. So this is how the electron is being transferred, okay. Electron is being transferred from one complex to another, okay. It is nothing but the that is the channels through which the electrons are being transferred, and the final thing what we need is, finally we just need the ATP, okay. So the end product is the ATP, that is energy molecule should be synthesized, and where it is synthesized? In the F0 and the F1 particle. Clear? So this is how the electron is being transferred between the various complexes in the mitochondria of the cell. Yes. So see, what are the various complexes? As I have said that complex 1, complex 2, complex 3, 4 and 5, right? These are the various complexes on the electron transport chain. So oxidative phosphorylation is what? It is a process in which energy is harnessed through a series of protein complexes. So what are this? What are this? These are all the protein complexes, okay? These are all what? These are all the protein complexes which will lead to the generation of ATP. ATP is finally harnessed with the help of this protein complexes embedded in the inner membrane of the mitochondria. So where does all these things taking place? It is taking place in the inner membrane, that is the cristae, right? It is taking place in the inner membrane of the mitochondria. Clear? Yes. So called the electron transport chain. So this chain is also called as electron transport chain and ATP synthesis. So finally what we need to create through the electron transport chain, finally we need to create what? ATP, okay? So inside the mitochondria, through this electron transport chain, what is being synthesized through this electron transport chain? Through this electron transport chain, ATP is being synthesized, okay? That's why mitochondria is also known as the powerhouse of the cell. So it is the synthesis of ATP by phosphorylation of ADP. So ADP is synthesized from the ADP, for which energy is obtained by electron transport and which takes place in the mitochondria during the aerobic respiration. So Krebs cycle is responsible for the aerobic respiration. Clear? Yes. Next, what is this oxidative phosphorylation? What is the purpose of this oxidative phosphorylation? So the oxidative phosphorylation is a highly efficient method of producing large amount of ATP. So what it is being used? Why it is being used? It is being used for the production of ATP, which is the basic unit of energy for the metabolic process, okay? So through this oxidative phosphorylation, the energy is being synthesized in the form of ATP, which is a basic process, okay, which is a basic process for metabolism. So during this process, electrons are exchanged between the molecules, which creates a chemical gradient that allows for the production of ATP. Okay, so there are various chemical reactions taking place which leads to the production of ATP. So you can note down these are the various complexes of the electron transport chain, that is complex 1. So what happens in the complex 1? So electrons from NADH, okay, electrons from NADH produced in the mitochondrial matrix during citric acid are oxidized as NADH dehydrogenase, and electrons are then transferred to ubiquinone located within the inner membrane, right? So what happens in the complex 1? The NADH, right? The NADH, from there electrons are transferred into the ubiquinone, right? This is Q, isn't it? Q means ubiquinone, which is sent to the inner, which is present in the inner membrane of the mitochondria. So electrons are being sent to the ubiquinone. So this is happening what? This is happening in the complex number 1. Now complex number 2, what happens? Ubiquinone also receives reducing equivalence via FADH2 that is generated during oxidation of succinate in the citric acid cycle. So ubiquinone is also being received from here, isn't it? As I have said that FADH2, which is released during the Krebs cycle, okay? So ubiquinone is also being released. Ubiquinone is also being released from the FADH2, right? From the complex number 2 in the inner membrane of mitochondria. Now this is happening in the complex number 2. So what is happening in the complex number 3? So the reduced ubiquinone is then oxidized within the transfer of electrons to cytochrome C via cytochrome B1 complex, right? So what happens? This reduced ubiquinone, so see this ubiquinone, okay, is being further sent to the cytochrome complex. So this is a cytochrome complex. So from the cytochrome complex, it is being, what happens from the cytochrome complex, it is being forming what? It is being forming, that is from cytochrome B to cytochrome C. So further through the cytochrome complex, it forms cytochrome A and cytochrome A3 in the complex number 4, okay? So see, in the complex number 4, okay, cytochrome C is what? It is a small protein which is attached to the outer surface of the inner membrane, which acts as a mobile carrier. So here, which one is the mobile carrier? It is cytochrome C. Okay, cytochrome C is a mobile carrier of the electrons, for transfer of electrons between complex number 3 and complex number 4. So here, as you can see, that cytochrome C is a mobile carrier. This is a mobile carrier which is transferring the electron between the complex number 3 and complex number 4. Okay, so cytochrome C is basically what? It is a mobile carrier of electron, which is transferring the electrons between cytochrome 3 complex number 3 and complex number 4. All right. So next, what happens in the complex number 4? So in the complex number 4, it refers to the cytochrome C oxidase complex containing cytochrome A and cytochrome 3. So further, the cytochrome C gets oxidized into cytochrome A and cytochrome C and two copper centers, and the electrons are then passed to complex 5 for the synthesis of ATP. So further, what happens in the complex number 4? The cytochrome C, okay, the cytochrome C, it gets oxidized into cytochrome A and cytochrome A3, isn't it? So further, it is being sent to what? It is being sent to the complex number 5, right? Complex number 5. Is that clear? Now what happens in the complex number 5? In the complex number 5, the ADP is converted into ATP, and with the help of enzyme ATP synthesis, right? So ATP is finally synthesized where? In the complex number 5. Then here, what are the two particles present? The two particles which are present in the complex number 5 are F0 particle and F1 particle, right? So this F0 and F1 particle, it finally helps in the conversion of the ADP to ATP with the help of the enzyme ATP synthesis, right? So this is happening in complex number 5. Next, when the electrons pass from one carrier to another via complex 1 to 5, in 4, in the electron transport chain, they are coupled to ATP synthesis, right, for the production of ATP from ADP and inorganic phosphates. So the energy which is released during the transport system is utilized in the synthesis of what? In the synthesis of ATP with the help of ATP synthesis. And this complex consists of two major compounds, that is F0 and F1. Clear? So what are the two major compounds of the complex 5? Complex 5 as F0 particle and F1 particle, where ATP is synthesized from ADP with the help of enzyme ATP synthesis. Is that clear? This is what happening in complex number 5. Okay. Now, what is this F1 complex or F1 compound and F0 compound? So F1 and F1 particle, F0 and F1 particle. So what are this? F1, which is the headpiece in the peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. So you have understood that in the inner membrane, that is the cristae, right? So here you have seen that various protein complexes, right? Like this you have studied in the mitochondria, right? That is F0 and headpiece is F0 and F1 particle, right? F0 and F1 particle. So these are responsible for the synthesis of ATP basically. So F0 is an integral membrane protein complex that forms the channel through which the protons crosses the inner membrane. So F0 basically helps in the transfer of protons. Okay, so see, this is the outer side. Okay, so this is attached to the cristae. Okay, this is basically the cristae. In the cristae only F0 and F1 particular present. So this is the inner mitochondrial membrane and this is the outside. So this is attached to the cristae. The headpiece is known as what? Headpiece is known as the F0, right? F1, sorry, the headpiece is known as the F1, okay? And F0 is the integral membrane part. Is that clear? So this is about complex number 5, okay? Now, let's see the molecules which are being synthesized in the electron transport chain. So you have seen that NADPH2, FADH2, these are the molecules. So NADH and I'm writing here NADH, FADH. These are the molecules which are being synthesized in the electron transport chain. So what are the molecules? The number of ATP molecule which is synthesized, it will depend on what? It will depend on the nature of the electron donor. Okay. So oxidation of one molecule of NADH gives rise to 3 molecule of ATP. So one molecule of NADH will produce 3 ATP molecule. Clear? One molecule of NADH, it produces how many ATP? 3 ATP. And one molecule of FADH2 produces 2 molecules of ATP. So one molecule of FADH2 produces 2 molecule of ATP in the electron transport chain. So last topic is the respiratory quotient. So what do you mean by this respiratory quotient? Okay, so whatever you have studied, right, that is the Krebs cycle, right, and the electron transport chain, that is, it is responsible for the plants, okay, how the respiration will take place. Okay, it is through which, through the electron transport chain, the ATP is being synthesized, because of which the respiration is taking place in case of plants. Okay, so next, the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed in the respiration is called as the respiratory ratio. Now it is denoted as the volume of carbon dioxide evolved by volume of oxygen consumed. Okay, and it is depending on the type of respiratory substrate used during respiration. This is nothing but the volume of carbon dioxide evolved by volume of oxygen consumed. Okay, this is respiratory quotient. So this is all about this chapter. So by this we are done with your chapter number 14. So I hope you have understood what is discussed in today's session. We have learned about the Krebs cycle and the oxidative phosphorylation, right? So if you find this video useful, do like it and also share it with your friends so that they find benefit out of it. So in my next class, I'll be starting with your chapter number 15, where you will be learning about the various plant growth hormones. Okay, till then, take care and bye-bye.